So here are my disclosures, none of which I think create any conflict, but I am going to talk about the research that I was doing at Stanford, as Julie mentioned. So gastrointestinal complications are important contributors to mortality and quality of life in cystic fibrosis. We've known for a long time that CF mortality is associated with poor nutrition. And more recently, through the Galaxy study and others like it, we've accumulated actual evidence that there is a very high GI burden in those with cystic fibrosis. And in this study, they found that up to a third of individuals had moderate to severe symptoms as assessed by a multitude of patient-reported outcome measures. Now, since we were able to see the effect of ETI on percent predicted FEV1, pulmonary exacerbations, and BMI, there's been strong interest in whether ETI may also have similar efficacy on the gastrointestinal symptoms of people with CF. This data from the PROMISE GI sub-study was published last year, and they looked at symptoms experienced by individuals through a variety of patient-reported outcome surveys, and this was after 6 months of ETI therapy. And what they noted was that there was a statistically significant decrease in GI symptoms. However, this did not meet the minimally clinically important difference threshold. They also noted that there was no change in stool frequency or consistency. And so there was disappointment from these results, I would say, across our community, and a lot of questions as to why. Subsequently, another similar type of study done with different questionnaires, the RECOVER study, this year published their results on patient-reported outcomes using different measures after 12 months of ETI therapy. And similarly, they saw a statistically significant decrease in symptoms, although with their scoring, it's unclear what is really clinically meaningful. But what I thought was interesting in looking at this data was, as you can hopefully appreciate, is that very rapidly after 1 to 2 months of ETI therapy, symptoms improved as indicated by a decrease here. But at 6 months, there seemed to be a rebound effect. And then later on at 12 months, depending on the symptomatology, there were varying effects. And it's unclear if these rebound effect at 6 months is due to physiologic factors or adherence factors. But I think it may help put into context some of the results from PROMISE and other studies that we're seeing. But nonetheless, more needs to be done in this area to understand how the GI complications of CF may be impacted by CFTR modulators. Now, CFTR is expressed throughout the gastrointestinal tract, and it is also expressed in many different cell types within the gastrointestinal tract, and this leads to complexity of symptoms. So CFTR is expressed in epithelial cells, enteric neurons, smooth muscle cells. It's also, although not shown here, in resident and migrating immune cells. And then even within epithelial cells, CFTR is expressed to varying degrees in different types of epithelial cells and different cells within the crypt villus axis. So what this means is that really we can see a myriad of symptoms because of the differential expression pattern across these different cell types. We can see decreased intraluminal pH within the intestine, which may impair pancreatic enzyme efficacy and impact the absorption of nutrients. This can impact mucus expansion and release. We can have decreased intraluminal fluid within the bowel, which can also contribute to compacted mucus and bulky stools. And then with the neuronal and muscle cells, we can also see decreased neural stimulation of contractions and hyperconstricted smooth muscle, all of which may contribute to gastroparesis, dysmotility, constipation, and incomplete evacuation. On top of all of that, we know that when you look at the intestinal microbiome of those with CF, there is a strong lack of diversity compared to individuals without CF. All of these different colors here in the non-CF bar indicate a number of different microbial species, and you can see a lack of that in the CF, and there's a decrease in richness and Shannon index that has been reported across a number of studies. Now, more recently, a group looked at the effect of long-term ETI on the intestinal microbiome. And here showing some of that data after a baseline at 0, 3 months of ETI, 6 months of ETI, and at least 17 months of ETI. And what they found was that they could see statistically significant changes, sometimes down, sometimes up, in the microbiome. However, what was I think really interesting is that when they compared that to healthy controls that didn't have CF, and that's on the bottom here, you can see that those changes were very small compared to what the intestinal microbiome of someone without CF looks like. So even if there are changes, they are small and may not have profound physiologic ramifications. I mentioned that gastrointestinal pH is important in digestive health, and it actually varies along the entire GI tract. And you can see that here in this graph that within the stomach, the pH is very low. And as you go into the first part of the small intestine, that pH rises in large part due to the secretion of bicarbonate secretion, and that will continue to rise throughout the small intestine. It decreases slightly as you go into the colon and then rises again. And when we've looked at what that looks like in individuals with CF, with different types of mutations, we can see that there are areas along the GI tract where that pH is impaired. It's more acidic. And for example, here in the proximal small intestine of the duodenum, the later small intestine, and also in some parts of the colon. Now, there are a variety of factors that contribute to gastrointestinal pH, including bacterial byproducts and proton transporters. But as I mentioned, epithelial bicarbonate secretion is an important contributor to that. In my lab, we used a variety of techniques to study intestinal bicarbonate secretion. We had animal studies, mostly in mice, and human studies involving endoscopic biopsies, tissue samples, and intestinal organoids. And using all of that, we performed short circuit current and pH stat measurements. We did transcriptomics and single cell RNA sequencing analysis, and used confocal microscopy as well, and put all these things together to try and understand better how bicarbonate secretion is regulated in health and also in CF. So we were very interested in a compound called linaclotide. Linaclotide is FDA approved for the treatment of constipation. It's a pharmaceutical analog of natural occurring peptides, guanylin and uroguanylin, and also a peptide made by E. coli called the heat stable enterotoxin of E. coli. And we were interested in linaclotide because we had previously shown that the heat stable enterotoxin of E. coli could stimulate CFTR independent bicarbonate secretion in the duodenum, or the proximal small intestine. And the studies I'm showing you here on the graph on my left are some mouse in vivo studies where we isolated the proximal duodenum, and we perfused through linaclotide in a sedated mouse at using different concentrations, and then measured the amount of bicarbonate that was secreted into the lumen of the intestine. And the dotted line here represents what the response to linaclotide alone. So at 10 to the minus 7 and 10 to the minus 5 molar of linaclotide, you see a nice increase in bicarbonate secretion. And we repeated these studies in the presence of a CFTR inhibitor, CFTR-INH172. And that data is here in the dark circles and solid line. And what you can see is that there's complete overlap of those responses. The CFTR inhibitor did not preclude the bicarbonate secretory response by linaclotide. We repeated these experiments using CFTR knockout mice and showed that there were similar bicarbonate secretory responses between wild type and knockout mice. Likewise, in human biopsies with and without CFTR INH172. And that when we looked at the short circuit current responses, we saw patterns that suggested involvement of chloride bicarbonate exchangers with no change in short circuit current upon linaclotide stimulation. Now, we were, based on that information, we were interested on what chloride bicarbonate exchangers could be involved in mediating this response. And we focused in on one called downregulated in adenoma, and found that linaclotide increases the apical brush border expression of this downregulated adenoma chloride bicarbonate exchanger. So here I'm showing sample confocal microscopy images to baseline, where you can see that there is a small amount of DRA at the apical surface, but upon linaclotide stimulation, you see that that increases, and that's quantified in the graphs below. We also saw that using duodenal enteroids from CF individuals that are F508 del homozygous, we saw a similar pattern of DRA trafficking to the brush border. Now, to see if these channels were functional, we repeated our mouse in vivo experiments with a selective DRA inhibitor, DRA250. And now the dotted line is this response I had shown you with CFTR inhibitor. And in the presence of the DRA inhibitor, now we can see that that response is significantly inhibited, suggesting that DRA is playing an important role in the CFTR-independent bicarbonate secretory response stimulated by linaclotide. So we put together this model where we believe that in the absence of functional CFTR, linaclotide can stimulate stimulate bicarbonate secretion through recruiting DRA to the apical membrane and activating it. Simultaneously, we also showed, although I didn't show the data here, that it can inhibit proton transport through sodium hydrogen exchanger. And collectively, this increases the pH out into the lumen. So the data I'm going to show you today is we had a question about, well, how does linaclotide increase membrane transport and activity of DRA, and could this mechanism be co-opted in CF to improve intestinal pH? So linaclotide, along with its other analogs, guanylin, uroguanylin, STA, are known to bind to guanylyl cyclase C receptor, and their canonical signaling mechanism is to increase intracellular cyclic GMP and decrease cyclic AMP, and collectively that stimulates CFTR-mediated transport and also inhibits sodium hydrogen exchange. But previously, we had looked to see if in CFTR knockout mice, we could see whether cyclic GMP could stimulate CFTR-independent bicarbonate secretion. And while we saw that 8-bromocyclic GMP, a cyclic GMP analog, could cause a robust increase in duodenal bicarbonate secretion in control wild-type mice, CFTR knockout mice did not have any response to that. And so that suggested to us that the responses that we are seeing in CFTR knockout mice and independent of CFTR may be occurring independent of this canonical cyclic GMP pathway. Now, there's old data that showed that the heat-stable enterotoxin of E. coli STA can increase intracellular IP3 and calcium, as shown here. So we wondered, could linaclotide also increase intracellular calcium and IP3 through phospholipase C activation? So to do this, we first measured intracellular calcium, and we did this by culturing human duodenal enteroids on cover slips and loading them with Fura red, an intracellular dye, which can measure changes in intracellular calcium. And I'm showing here the average response to multiple enteroids in black, and the gray shading is the standard air. And what you can see is when we treat these enteroids with linaclotide at 10 to the -7, the same doses we used in our prior studies, we can see a rapid increase in intracellular calcium, but it's transient and it returns down to near baseline. For comparison, when you give carbachol afterwards, you can see a similar type of increase, although its pattern is different. And when we quantify this, we see that linaclotide can actually stimulate a larger peak increase in intracellular calcium than carbachol, although again, it's not as sustained as carbachol. So next we looked at PLC inhibition, and to do that, we used a compound called U73122, which is specific for to inhibit PLC. And these studies are, again, those in vivo bicarbonate secretion studies in mice. The first set of data that you have here in the clear bars is the data I showed you previously, just in bar graph form, where we have an increase in bicarbonate secretion at 10 to the -7 and 10 to the -5. Now, when we do the same types of experiments with U73122, here in the gray bars, you can see we no longer see an increase in linaclotide-stimulated bicarbonate secretion, suggesting that PLC is involved in the CFTR-independent bicarbonate secretory response. So we next looked at whether PLC was involved in DRA trafficking. So again, these are apical out human duodenal enteroids, as I showed you before. We have staining for now DRA in the green, actin to mark the plasma membrane in red, and then merged pictures across 3 different conditions here: water control, linaclotide stimulation, and then linaclotide stimulation in the presence of this PLC inhibitor. And they've quantified these in the graph to the right. What you can see is in the presence of linaclotide alone, we see an increase in membrane expression of DRA, as I showed you previously. But when we do the experiments in the presence of this PLC inhibitor, we can completely block that movement of DRA to the membrane, again suggesting that PLC is involved in mediating DRA trafficking to the membrane when stimulated by linaclotide. So we became interested in a protein called IRBIT, and IRBIT is an acronym for a long name called the IP3 receptor binding protein released with inositol 1,4,5 triphosphate. You can see why we call it IRBIT and don't say that all the time. And its gene name is AHCYL. Now, IRBIT's a hub protein. It's been known to be involved in facilitating the transport of proteins to the cell membrane, and some of those here, which people may recognize, in the pancreas. However, it's been unknown what role, if any, IRBIT could play in duodenal ion transport. And given what we had seen with PLC activation, then we thought it was an interesting protein to look at, because what happens is normally IRBIT is bind to IP3 receptors on the endoplasmic reticulum, and upon PLC activation, where you get an increase in IP3, IP3 can bind to a similar place on the IP3 receptor and actually displaces IRBIT, which can bind to transporters that may be localized within the cell or at the membrane and can either activate them or help traffic them to the membrane. And so this was our hypothesis that normally we have this sort of pool of DRA that's located within the cell, and IRBIT's bound to IP3 receptor, and upon linaclotide stimulating PLC, the IRBIT is released, and that can recruit this intracellular pool of DRA to the membrane for bicarbonate secretion. We first looked at Protein Atlas to see what the distribution of IRBIT might be in the intestine and the duodenum specifically. Of note, there are two different types of IRBIT. There's IRBIT encoded by the HCYL1 gene, and also long IRBIT encoded by HCYL2 gene. And I'll just draw your attention to GI tract, and hopefully you can appreciate that they seem to have different expression patterns, with the HCYL1 more dominant in the duodenum and the HCYL2 more in the stomach and rectum. So for our purposes, as we were interested in what was happening in the duodenum, we saw that HCYL1 was high and HCYL2 was low. We confirmed these results from our own analysis of existing single-cell RNA sequencing studies. When we examined one set of data, we found that there was robust expression of HCYL1 in the duodenum, but could not find any expression of 2. When we looked in another data set, we could see expression of 1 and 2, although 1 was expressed at a higher level and in more cells. When we looked at a third data set that had another segment of the small intestine, the ileum, we saw a similar pattern where HCYL1 was expressed higher than HCYL2. But as you go to other parts of the GI tract, the colon, for example, that started to shift, where actually these expression patterns looked a little bit more similar. And then when you go to the rectum, actually there's a reversal where HCYL2 seems to be greater than HCYL1. And so it depends on where we're looking. But since we were interested in the duodenum, we focused mostly on HCYL1. Now, as you can see, most of the IRBIT expression here is in the enterocytes. And so we wondered, well, how is that expressed along the crypt-villus axis? And so using the duodenal enteroids that are differentiated to differing degrees, which allows you to create more crypt-like and villus-like enteroids, we saw that as you differentiated enteroids, the IRBIT expression increased, suggesting that IRBIT expression may be higher in more villus-like cells. Interestingly, it also followed a similar pattern as DRA. And we looked at protein expression using immunofluorescence, and this is a human duodenal biopsy taken during endoscopy with IRBIT in red. You can see that the greatest amount of IRBIT expression seems to be in sort of the mid to upper villus regions, and less so down in the crypts. So IRBIT is definitely expressed in the duodenum, and we began to characterize where. We also use this RNA sequencing analysis to see if the mRNA is co-localized with DRA or SLC26A3 in the same cell types, and crypt samples and also villus samples. And I'll just draw your attention to the upper right corner are those cells that are double positive for both DRA and IRBIT. And we can see that in both crypt enterocytes and villus enterocytes, they have similar amounts of colocalization, although they differ in cells that only express IRBIT, but not DRA, and that's this lower bottom corner. When we look at protein colocalization using confocal microscopy, we do see areas where DRA and IRBIT colocalize. The top set of images are from sections of human duodenal biopsies, and the bottom set is through whole mount microscopy. And I've included arrows here where you can see we're looking for the areas that the green and the red overlap to create this yellow color. And you can see there are different areas on the villus regions where both DRA and IRBIT colocalize, and there are regions where they don't. And you can see that also here on the whole mount microscopy. So this all just suggests that there may be some relationship with DRA and IRBIT. So we wanted to see, well, what happens to IRBIT localization when we give linaclotide? So in healthy enteroids, we performed similar experiments with water control and linaclotide stimulation. And under baseline conditions, we see IRBIT here stained in red and the membrane in green by Villain. That IRBIT is mostly intracellular, not much at the membrane. But when you stimulate with linaclotide, most of the IRBIT then traffics to the apical pole, and some of it goes within the brush border, as you can see this yellow overlay. And that's quantified here in the graph to the right, where we see a significant increase in the membrane localization of IRBIT. We repeated the same experiments in CF enteroids, and we saw a similar pattern of distribution where there was increased IRBIT at the apical membrane. When we compared the mean fluorescence intensities between healthy enteroids and CF enteroids, there was no difference. However, due to some subtle differences in baseline membrane expression of IRBIT, when we looked control to its own self here with and without linaclotide, we see that there actually is a little bit less of a magnitude of stimulation in the CF enteroids than healthy enteroids, and that's likely because there's a little bit more IRBIT at the membrane during non-stimulatory conditions in CF enteroids. So then we wanted to see, well, what happens to IRBIT localization with the PLC inhibitors. And so similar experiments I had shown you with DRA, we looked at IRBIT localization with U73122. And what we saw is that with linaclotide, you continued to get an increase in the membrane localization of IRBIT, even with PLC inhibition. However, when we again normalized these to their respective controls, we did see that there was a reduction in the amount of IRBIT that was going to the membrane, suggesting that linaclotide does not only stimulate IRBIT to the membrane, but it does so at least in part, but not totally through PLC. So in summary, linaclotide does stimulate apical trafficking of DRA, and I think we've shown that, well, that it involves PLC signaling. IRBIT, which is a downstream effector of PLC, is expressed in a similar pattern as DRA and healthy and CF enteroids. And like DRA, linaclotide can also stimulate IRBIT to traffic to the apical pole. And PLC inhibition partially, but not completely, decreases linaclotide-stimulated IRBIT trafficking, again suggesting that IRBIT may utilize non-PLC mechanisms for trafficking. It's unclear if these are also involved in DRA trafficking or some of the other cellular functions that IRBIT may have. So we've shown that IRBIT may be involved in DRA trafficking, but I think there are more definitive studies that are necessary to confirm this. Thank you. And lastly, I just want to recognize those people that have really done the work. I want to highlight Dr. Jessica Sarthy, who in the majority of the experiments that I talked about today, and also the other people that were in my lab that contributed to these projects, as well as collaborations with the Quo Lab at Stanford and labs at UCSF and Cedars-Sinai, and then the people that funded the work, and a special thank you to CFRI.